into the membrane to destabilize the lipid bilayer packing, and then that creates pores, temporary transient pore formation that larger molecules can exploit then and transport. And then there's a carrier monomer here by which you can add in proteins, oligonucleotides, siRNA, and I'll show you an example of that. We've shown in a lot of studies, this is a recent illustration, that these polymers do have this property of a pH-dependent membrane destabilizing activity that is reversible. And so in this assay, this is an assay that's been used for many years in the field, a standard assay, standard conditions that allows you to compare different compositions across the literature. And it's an outside-in membrane destabilizing activity. You add polymers to a suspension of red blood cells, and then as a function of pH, ask the question, how effective are they at destabilizing that membrane sufficiently for hemoglobin to come out of the cell, which is what you actually measure here on the y-axis. It's called hemolysis. And so I'll just show you that at these compositions, at pH 7.4 physiological pH, you have very low membrane destabilizing activity. But by the time with this composition you're up at or down to pH 5.8, this polymer is converted into a membrane destabilizing form. And we've done a lot of work. You can very nicely tune exactly what pH this occurs. We have polymer compositions that will destabilize at pH 6.6, for example, and not at 7.4. So these are activated extremely sharply, very similar to the biological proteins across a pH range of less than a half a pH unit, and it's fully reversible. So the idea then is that we might, for example, through this monomer add a cationic graph that condenses either negatively charged plasmid, negatively charged oligonucleotide, or negatively charged siRNA. And the active form then is a polyplex, or a condensed particle. And the details of size and charge are very important, and they can be controlled very nicely in a rational way by the molecular weight and the composition here of the polymer. But just to emphasize, these are particles. This is one composition, dynamic light scattering studies, to show you that the size range is around 200 nanometers for this particular condensed form. But just to emphasize that these do form particles. I'd like to just give you one application, and that is in the tissue regeneration area, is control of the foreign body reaction. Shown here, the response to a scaffold. There's been a lot of discussion of this. And we've been very interested through UWeb with Buddy in trying to control the foreign body reaction by the nucleic acid approach. And our approach targets the macrophage, which has been an incredibly difficult cell to get nucleic acids to, because it has a form of the lysosome that's generally called the phagosome that is particularly rich in these nucleic acid degrading enzymes. What we're after is signaling pathway modulation, so not to simply knock out the macrophage, but to very specifically control the activation state of the macrophage. You don't want to knock it out. There has to be some inflammation so that you can enter into wound healing. And so we've been conceptually interested in the idea that you can, in a time-dependent way, knock out very specific signaling pathways that are associated with the macrophage activation. And let me show you the exact composition here. Similar composition, there's a membrane-disruptive polymer backbone that is pH responsive. In this case, we've just grafted on through a disulfide linkage a polylysine segment that electrostatically condenses antisense oligonucleotides or siRNA drugs. And this disulfide allows the carrier to separate from the drug in the cytosol. The key new thing, really, again, just to emphasize, is that the polymer has an activity. It's not a simple carrier. The target that I'll show you some data for, the mRNA target, is interleukin-1 receptor-associated kinase. This is a key kinase that is constitutively expressed and is up very near the toll receptor pathways, but also the IL-1 beta pathway, which is very important for the foreign body response to biomaterials. And so the idea is to knock this out, at least transiently, by delivery of an antisense or siRNA against that IRAK mRNA. This is IRAK1 for the detail. But essentially what you do by knocking out IRAK is short-circuit the macrophage's ability to receive pro-inflammatory signals. And you can measure that if you, for example, challenge macrophages that have been treated with your drug with LPS. Is it true that, for example, you lose the connection to the upregulation of TNF-alpha? We've shown that. I'm just going to show an siRNA result. We can do this in vitro, sort of, with any number of different antisense and siRNA drugs. This is kind of a typical result. Here, what I'm actually looking at, rather than TNF expression, is by RT-PCR actual IRAK message levels. So if you have no carrier, this particular siRNA has no activity in the macrophage. And we just found that to be generally true. It's hard to get nucleic acids into that target cell. This is a dose response then as we're delivering with this pH-responsive carrier, and you can see a very nice knockdown in a dose-dependent way. And you can do a similar study, maybe not as effective. It is, in our hands, we've seen better siRNA results than with the corresponding antisense. And I'm not going to go into details, but we actually have had these in a number of animal models now and shown, for example, in a wound healing model at UWeb, that these types of pH-responsive carriers are really, it's kind of an all-or-nothing approach in this local delivery system with a plasmid. And they've also been in a lot of systemic delivery models to look at toxicity and biodistributions. And they pass sort of the first test, but there's really a lot more preclinical validation that needs to occur in specific applications. But I give this sort of as a specific example. I could show you several others from different groups. It's an area that's very active to try to build more active polymeric delivery systems. And I use that to illustrate the point that what people are after at this point is something that goes beyond a simple carrier, because those just have been shown to be insufficient in the clinic. And so these new delivery systems have some sort of activity to get around these barriers. And in our case, the primary technology is endosomal escape and this property of the polymer itself in a pH-dependent way being membrane destabilizing. But of course, in addition, they have the usual advantages to condensing, to providing serum stability. I didn't really point that out so much, but these polymers also provide a lot of serum stability so that compared to most cationic lipids, they show much higher activity in the presence of serum. And so for in vivo applications, this is a key point, obviously. So with that, I'll just close. I guess maybe you can see, I was thinking this morning when I was looking at this, this is our pyramid, I guess, in Seattle. It's called Mount Rainier. The disadvantage of our pyramid is that it can blow up at any second. So I don't think it's quite as stable as your pyramids seem to be. But thanks for your attention, and thank these people over here for funding.
Thank you, Pat. This presentation is open to questions.
May I ask a basic and probably naive question? There has been here a new elegant lecture about the modeling of the nucleic acids. I mean, it's a crucial point in non-viral delivery systems. On the other hand, routine modeling has been the floor of considerable progress regarding the software. How far is we are in the area of modeling of nucleic acids. Can you elaborate on this, the software, how the three-dimensional configuration of the DNA or the RNA?
Well, I can't answer it with any detail. I mean, to my level of knowledge, the structure of DNA is so well defined conformationally that it is, in my mind, it's pretty well described. With siRNA as well, there's some initial high-resolution structure analysis that have been done on some of those forms, and they adopt typical nucleic acid, helical conformations. But when you're maybe